Hello students, let us study about the parts of a microscope. So as we can see the picture, it is a picture of a compound microscope and our objective is to learn the construction and working of a compound microscope. A compound microscope has multiple lenses which collect light from the object and then focus the light into the eye. Microscopes have the ability to magnify very minute objects to a substantial size, clearly showing the smallest details. So there are three major parts of a compound microscope: a body tube, an objective lens, and an eyepiece. Now the body tube is composed of two smaller telescoping tubes with lenses coaxial mounted at either end. So there is a wider tube and a narrow tube. The wider tube holds the objective lens and the narrow tube has the eyepiece. The knobs for both coarse and fine adjustments. They both allow the tubes to slide onto one another, varying the distance between the two lenses. Now the lenses are convex with different focal lengths. The lens closer to the object is the objective and has a short focal length, usually below 1 centimeter. The lens closer to mirror the eye is the eyepiece, although eyepiece has a shorter focal length. So the above lens is the eyepiece and below lens is the objective. Then there is a revolving, as you can see there is a revolving nose piece which is also known as a turret. This is the revolving nose piece attached to the lower end of the objective. It can be rotated to change the magnification as necessary. Now below the objective there is a flat platform which is known as a stage, and it is with two clips that holds the slide in the place. Now as you can see, there is a small aperture in the stage allowing light into the body. Now the body tube is arranged on an arm which is connected to the base that allows microscope to tilt a little bit. Then there is a small mirror which is present at the base of the microscope that reflects the light through the stage aperture above. Above the mirror you can see an iris or a diaphragm is present, which helps in controlling the amount of light passing into the stage aperture. Now let us study the working of a compound microscope. So the object is placed just outside the focus of the objective lens, and we can see a real, inverted and a magnified image is formed, which is designated as I1. So this is the first image which is formed, I1. Now the magnified image, this I1, it acts as an object for the eye lens. The eyepiece is adjusted such that the image I1 lies within the focus of the eyepiece, that is FE. So the eyepiece acts as a magnifying glass and it forms a virtual, erect and magnified image I2 of the object. Now the magnifying power of a lens is expressed in diameter. So if we want to calculate the total magnification power of a microscope, so the total magnification power of a microscope M is the product of magnifying power of the objective M0 into magnifying power of the eyepiece. So for example, if the objective lens has a magnification of 40x and eyepiece has magnification of 10x, so the total magnifying power of the microscope will be 400x. So let us summarize the topic. The compound microscope has a system of convex lenses allowing the magnification of its minute objects to that of a very large image.